The scientific method may have been taught to you as a pretty linear procedure. Maybe you were in elementary school, you had to do science fair projects, and they gave you a list of steps, and they said, this is the scientific method, and you have to do it this way. In reality, the scientific method is a little bit more flexible. So that list of steps is a great guideline, and we'll go through it. The steps are in this diagram here. So make observations about the world, think of a research question, come up with a hypothesis, which is like a proposed explanation, develop a prediction as to if you do your experiment, what do you think will happen? Do your experiment and gather data to test whether your prediction was accurate. And based on that data, you may go back and change your hypothesis and make a new prediction and then gather data again and see if anything changed. And then once you've gathered your data from your experiments, you might develop a general theory that might explain the phenomena that you're observing. So again, even this is a guideline, not like a strict rule, but just remember that the scientific method is constantly ongoing. It's not just step A to step Z and then you stop. So we're going to go through the steps of the scientific method with a hypothetical experiment related to something that was in the news over the weekend. So I made the observation that people are making homemade face masks to stop the spread of the new coronavirus. You may have seen this. I think Donald Trump suggested that people wear face masks this weekend. And the idea of these homemade face masks is not so much to keep the virus out, like virus in the world from infecting you. It's to keep you from spreading the virus if you are an asymptomatic carrier, for example. So my mom wants to make, this is true, my mom wants to make some of these face masks and give them away. And so we were talking about what is the best design, what is the best material to use to make these things actually effective at preventing the spread of the virus. So that's my research question for this theoretical experiment. What type of face mask material is the most effective at blocking the passage of the new coronavirus through the mask? And I'm going to come up with a hypothesis. So a hypothesis is a possible explanation of how something works. You can kind of think of it as like a potential answer to your research question. It's an educated guess. So if my question was what material is the most effective, then my hypothesis, let's say thick face mask material is the most effective at blocking the passage of the new coronavirus. And a note here, science is not about proving your hypothesis right. It's totally okay if your hypothesis is wrong. It's more about just having a jumping off point, like somewhere to start with. So if you get data that doesn't match your hypothesis, there is nothing wrong with that. And you should never go back and say your hypothesis was different so that it matches your data, because it doesn't matter. Your hypothesis doesn't have to match your data. A lot of times we learn things when we get unexpected results. And one thing you definitely never ever want to do is change your data to match your hypothesis. That's very unethical. And then a note about a good hypothesis, it should be testable and falsifiable. So our hypothesis that thick fabric prevents the spread of the virus through the mask, we can test that by, say, trying to spray virus particles through the mask, like a pretend sneeze, and see how many particles come through. And if it doesn't do better in thick masks, then we falsified our hypothesis. So any hypothesis that you can't do a test for and you can't try to disprove is not an appropriate hypothesis for a scientific experiment. So the hypothesis that magical green fairies make the plants in the forest grow, we have no way of testing that, and so it's not an appropriate scientific hypothesis. Okay, so I have my hypothesis. Sometimes you'll see predictions as the next step after hypothesis, but I actually think it's important to come up with your experimental procedure before you make a prediction, because you can't know what to predict if you don't know what your experiment's going to look like. So here's the experimental procedure that I've designed for this experiment. So I'm thinking, okay, people need to make these masks from materials they have around the house. So what are some materials that might be in clothes? So let's see, there's silk, there's like a thin cotton, like a t-shirt cotton, there's flannel, there's fleece. So I'm sure there's more, but those are four that have different thicknesses. So then I'm going to, actually I'll show you the picture. So I'm going to take a spray bottle of virus particles mixed with water in this pretend highly controlled lab experiment where everybody's protected from the virus, and for some reason I have virus particles available. I'm going to take a clear plastic tube with the mask material attached to the end, and I'm going to spray the virus, like a set amount of the virus, through the mask material, and I'm going to take a little cotton swab and I'm going to measure how much of the virus falls down onto the lab bench on the other side in a set area of 10 by 10 centimeters. So in these trials, you want to make sure that everything is the same to the best of your ability, except for the one thing you want to change. So in this case, the thickness of the fabric. And one thing I want to point out with this experimental design is it's actually not a perfect experimental design because the thickness isn't the only thing about these fabrics that differ. Silk is made from animal proteins from the silkworm, cotton's made from plant fibers, so our fabrics don't just differ in thickness, so it's actually not a perfect experimental design the way that I've set it up. One thing that we talk about a lot that it can be a little hard to wrap your mind around, but you'll get it over time, is the idea of independent versus dependent variables. So an independent variable in an experiment is the thing that the scientist is changing. So in our case, it's going to be the thickness of the mask material is what we are changing between each trial. And the dependent variable is the thing that depends on the independent variable, and so it changes as a result of the independent variable changing. You can think of it as the thing that we're measuring. So in our case, if the independent variable is the thickness of the mask, we think that how much virus makes it through the mask depends on the thickness of the mask material. So that is our dependent variable. And then there are a couple more things that we want to throw into our experiment to make sure it's really thorough and complete. So we need experimental controls. Controls are experimental trials, and what trials are will start to make sense as we proceed through the course and do more experiments. But controls are like experimental trials where you already know what results to expect. So a positive control is something you put in your experiment where you expect to get positive results. For example, if we used an N95 mask in our experiment, we already know from previous tests that N95 masks block about 95% of virus particle transmission, something like that. And I could be wrong on that number, but it's a high number. And a negative control, you expect to get negative results. So for a negative control, we could use no mask. We know that we expect no mask not to block any viral particles. And so if we, do our spray test, use our cotton swab on the bench, and we aren't getting lots and lots of viral particles on that bench, then maybe our method for detecting the number of viral particles on the bench is not working properly. So you use your positive and negative controls to make sure that when you compare your results from your other tests to those controls, that they fall within the right range that you would expect to see. And it also helps you make sure that your experiment is working. And if your experiment isn't working, it can help you figure out which part is wrong. Okay, so we've got our experiment designed, so let's make a prediction. A prediction is similar to a hypothesis, but it's more specific, and it's specific to your experiment. It's based on your hypothesis, and it can be worded as an if-then statement, but it doesn't have to be. So our hypothesis was that thick face mask material will block the virus. Our prediction is going to be, again, specific to our experiment. When we spray viral particles through fabrics of different thicknesses, thicker fabrics will allow a lower percentage of viral particles through. And we could have written this as if-then. We could have said, if we spray viral particles, then thicker fabrics will allow. But you don't have to. I just wanted to show it to you both ways. So now that we've made our prediction, we're going to actually do our experiment that we designed. We're going to record our observations, and those the things that we're recording are called data. One is a datum, multiple are data. And data fall into two categories. You can have quantitative data, which is numerical data. For example, we're going to be recording the number or maybe percent of viral particles that are found in that area of the lab bench. There's also qualitative data, which we won't be using in this experiment, but it is descriptive data. So if Jane Goodall observed chimpanzee behavior and like drew pictures and stuff and just described it, then that's descriptive qualitative data. Okay, so we've done our experiment and we've collected our data of, let's say, what percent of the virus that we sprayed into the mask came out the other side and landed on the table. Once you have your data, you analyze your data. And what you're often looking for are trends or patterns in the data. And to help you do this, you put that data in visual tools a lot of the time. So tables, graphs, charts, illustrations, things like that. We talked about independent and dependent variables. When you are graphing your data, you put your independent variable, the thing that the researcher chose to change, along the x-axis or the horizontal axis. You put your dependent variable, so the thing that changes as in response to the thing that the researcher wanted to change. So in our case, that would be the mask thickness is independent on the bottom, and the viral particles that made it through is dependent on the left-hand side there. So you put your dependent variable on the y-axis or the vertical axis. So here's a pretend graph of pretend results of our experiment. So again, thickness, I put the mask materials in order of imaginary thickness. I don't know if silk is thinner than N95 masks or not, but I just put it that way. And then virus particle transmission on the left-hand side, meaning if 100% of the virus particles made it through the mask or absence of mask onto the lab bench, then you have a high score. And if closer to 0 means that the mask blocked almost all of the virus particles from getting through. So in the beginning of this graph, it almost looks like our pattern is working, right? So no mask would be like infinitely thin, was the highest transmission of viral particles. Silk blocked a little more than no mask. And then N95, which we're pretending is slightly thicker than silk, blocked a ton. So it looks like our hypothesis is true. But then we jump over to the cottony materials. So the thin cotton didn't block very much. The flannel blocked a little more, and the fleece blocked a little more than that. So it kind of looks on the right-hand side of the graph like the thickness of the fabric matters too. So we might have something here, but it's not a strict trend. So now that we have analyzed our data, we can draw conclusions. And drawing conclusions involves developing probable explanations for the results that we observed. Basically, we have this data. What does that data tell us? What does it mean? In our case, in that graph we just looked at, I might say, well, it looks like the thickness of the fabric may have some relationship with how effective the fabric is at blocking the virus, but it is not the only thing that explains how effective the material is. So we need to look for other factors that help make the mask effective at blocking the spread of the virus. And so once I have that conclusion, I'm going to develop a new hypothesis. Like, if it isn't thickness, what might it be? So in this case, maybe my next experiment would be face mask material with tiny pores is the most effective at blocking the virus. And then I could set up a new experiment where I measured the size of the pores in the fabric and tried spraying the virus through it again and determined whether the small pores affect the viral transmission through the mask. Sorry, dog. So that is it on the scientific method. You will be using the scientific method throughout the quarter. So we will get to talk about this lots and lots. That's it. Thanks, everyone.